Hi there, today we're talking about the different extreme environments where you can find bacteria. Extremophiles are organisms that live in conditions so extreme that only a few species can survive. These conditions could be extremely hot or cold, acidic or alkaline, and even desiccation areas. Did you know there is a species of bacteria called Thermochronis ruber that survive in thermal vents at Yellowstone National Park? They can grow under temperatures ranging up to 89 degrees Celsius. This classification of heat-loving bacteria are called thermophiles, and they can survive in temperatures between 45 to 80 degrees Celsius. Organisms that grow between 80 to 122 degrees Celsius are called hyperthermophiles. Thermophiles can survive in these extremely hot environments because of the unique sets of molecular adaptations. They have a higher melting point due to their membrane lipids containing longer-chain fatty acid. Recently, it has been discovered that an increase in basic amino acid at certain positions causes more ionic bonds, and this increase in ionic and hydrogen bond interactions results in increased thermal stability. Enzymes play an important role in the way thermophiles can grow in extremely hot environments too. For example, thermophilic lactic acid dehydrogenases is a thermophilic enzyme that is less flexible, resulting in a less efficient catalyst at lower temperatures. Speaking of lower temperatures, bacteria can also grow in extremely cold environments, and these bacteria are called psychrophiles. Psychrophiles can grow and reproduce between temperatures lower than 20 degrees Celsius. They are found in environments that are permanently cold, so think of deep ocean waters or the polar ice. Psychromonas ingrahami is a bacteria found to grow at sub-freezing temperatures of negative 12 degrees Celsius. How can they survive in such cold environments? Well, firstly, to prevent them from stiffening up, they have a lipid cell membrane made up of high amount of short, unsaturated fatty acids. Psychrophilic bacteria also have transcriptional and translational enzymes that adapted to be optimally active at lower temperatures. This plays an important role in protein synthesis. The enzymes in psychrophiles are much more flexible, and this compensates for the freezing effect. To increase flexibility, the enzymes generally have reduced number of ion pairs, hydrogen bonds, and hydrophobic interactions. The other unique characteristic of psychrophiles is that they have proteins called antifreeze proteins, AFPs. AFPs bind to ice crystals and prevent the growth of small internal ice crystals in organisms.
Extremophiles that inhabit acidic or alkaline environment are respectively called acidophile and alkalophiles. The subgroup of microorganism is able to keep the internal cellular environment with a neutral pH of 7, nevertheless the outside environment. This is possible thanks to different active and passive mechanisms used by the microorganism. An acidophile lives and survives in an environment with a pH ranging from 1 to 5. They inhabit sulfuric pools, geysers, and the stomach of animals, which has a pH of 2. Acidophiles are able to maintain the internal environment neutral by an active pumping of hydrogen ions from the cell. This maintains the internal pH and makes the outside acidic. The subclass of extremophile is indeed of particular interest for the scientific community, as the environment in which they live is a consequence of the cellular activity instead of being a condition dictated by the environment itself. Examples of bacteria acting on acidophiles are Acetobacteria aceti, which makes acetic acid, vinegar, by oxidation of ethanol, and Alicilius bacillus, which is a bacteria that can contaminate fruit juice that has a pH of 3.3. The counterpart of acidophiles are alkalophiles. These organisms live in extreme alkaline environment with a pH ranging from 8 to 11. This particular condition are found in the Rift Valley in Africa and in the Soda Lakes across the United States. Alkalophile as acidophile use both active and passive mechanisms to keep the intracellular matrix neutral. An example of passive control is the presence of acidic polymer in the cell wall. Those polymers are able to control and diminish the amount of hydroxyl ions diffused in the cell. Examples of alkalophile bacteria are the Anabena and the Microcystis, which is a genus belonging to the cyanobacteria.
Whose most halophiles are classified into the archaea domain, there are also plenty of bacterial halophiles. Halophiles are able to be found in places that have a concentration of salt 5 times greater than that of the ocean. These places, such as the Great Salt Lake in Utah, or even the Dead Sea in the Jordan Rift Valley. In terms of the classification, they are best suited to be categorized as mild, moderate, or extreme halophiles. Mild halophiles are able to tolerate a salt concentration of up to 6%, whereas extreme halophiles have the ability to withstand salt concentrations of up to 30%. Some examples of halophiles are the Halococcus and the Halobacterium salinarium. In order for halophiles to survive in such conditions, they are required to prevent desiccation through the osmotic movement of water out of their cytoplasm, or they are accumulated from the environment into the cytoplasm. These organic compounds are known as osmoprotectants. Now, in terms of a desiccated environment, some extremophiles have the ability to withstand a desiccated environment for a prolonged period of time. To desiccate means to remove the moisture from an organism or an object that normally contains moisture. In such an air-dried state, some bacteria have the ability to survive for only a few seconds, whereas other bacteria have the ability to tolerate desiccation for a longer time period, such as the bacterium H. marinus. A desiccated cell is distinguished by its lack of water, with contents as low as 0.02 grams in dry weight of H2O. At a level such as this, certain monolayer coverage by water of macromolecules, which include DNA and proteins, is disturbed. There isn't any evidence of obvious adaptations of proteins that can encounter the effects of air drying. However, some cyanobacteria have marked the ability to do so. Bacterium features, such as the elaboration of an extracellular glycan and the synthesis of a certain UV-absorbing pigments, help contribute to the bacteria cell's maintenance of structural integrity and protein stability.
So, acidophiles, alkalophiles, thermophiles, psychrophiles, and halophiles are just some of the examples of extremophiles that you can find in extreme environments. We hope you've enjoyed this video and thank you for watching.